Hey, all right, we got it.
Okay. All right. So great. I stayed up late last night putting these slides together. Didn't have a ton of time to prepare, so hopefully they're clear. Uh, if you have questions or want to dig in, go ahead. I think I have about 45 minutes worth of slides probably. But there's a lot of there's probably 4 hours worth of information in these papers that I could cover. So if we do want to dig into any of especially the bio side of things, I can describe a lot of what's going on in more detail. But I focus mostly on the machine learning stuff, but I'm going to give a little bit of genetics two slide introduction to genetics just for people who don't have experience with it. Um, okay, great. So, whoops, why is it not? Okay, so this is sort of the why. So the Evo is a foundation model. It's like in a series of there was like hyena DNA, and then there was Evo, and then Evo 2. It's a 7B and 40B model. And the purpose of a model like this is so it's nucleotide based, meaning it's actually modeling the DNA sequences. And the purpose, these are some of the applications on this slide for that. So like there's a lot of prediction things where you can like for just like I'll go through these, maybe not all of them, but I'll go through a lot of them. So like if I knock out genes, is it important? Does it impact, does it impact like the health of the person or the function of something, or does it not matter, or like a portion of the gene? What this one is actually really, really important and hard to do. So they seem to have made progress. I don't know enough about the biology to know how realistic what they're saying they're claiming is and how well they've tested it. But being able to predict the effect of your genetic variations on like drug treatments, your health, other things like that, um, is sort of the precision medicine promise, and it's quite difficult. But this kind of model seems to be helpful for that. Like designing genome scale things, I don't really understand that use case super well in terms of practical things, maybe for study. Um, but looking at predicting like sort of protein mutations based on gene changes, sort of things in the protein, like what feature, like identifying structures in the feature, structures in protein folds, uh, sort of designing proteins, predicting, sort of like in the RNA, like being able to find mutations, sort of predict whether a RNA strand will be stable or not, sort of like, and then things in the epigenome. Epigenome is like how is other physical mechanisms that cause genes to be more or less expressed. And so, and chromatin is like a major, one of the most important things with that. And so, like sort of making predictions about that. So, like all of these things, they have these really either really important scientific or sort of scientific consequences, like being able to understand or monitor or measure things better, or they actually have impact on your ability to sort of make clinical decisions with. So I think so. There's like doing this well is exciting, and it does seem like this does seem like a bit of a step change to me with not knowing a whole lot. There's been a lot of attempts at models like this and they do work, but this seems to, because it's well funded and done by a large team and, sort of like has support from NVIDIA and other things, it seems to be like a pretty impressive effort. Um, so this is the interesting thing about Evo is that this is not just for human genome. This is for, so they incorporated genome. So each point on this UMAP is a separate genome, and or part of it, at least part of a separate genome. And so that you can see there's like a uh, ton of different genomes here. And I think this core data, that's where humans would be, somewhere in there. So so you can see, and that's, I guess, this one probably. So this area. So like you can see that it's, that there's a lot of other stuff that got included. And that the purpose of that is to be able to transfer your understanding of the behavior of genetics from these other species into humans, and also to study those other species. Okay, is that clear before I dive into this? Please interrupt if you guys have questions. Okay, so this is, I say probably wrong because this is just my pretty lay understanding based on like, college biology and uh, conversations with computational biologists and things like that. So, but I think that for AI engineers, we don't need to understand it too well. There's like sort of so I'm going to give a conceptual view and then maybe one level deeper. That so there's genetics kind of deals with these three factors, and then there's lots of other stuff that is related, but DNA, RNA, and then proteins. So, and then proteins turn into larger structures and signaling and all sorts of stuff. But sort of like at its core, at least from my understanding, um, uh, you sort of have DNA, and this is like the long-term storage, and and I like to think about this as the software, right? Like this is the program that your cells follow in order to create proteins, which are sort of like hardware and sort of like software. So like either, and you could call it software or RTL. So like you're, describing how to synthesize a piece of hardware maybe. And then RNA is a less stable, kind of very similar to DNA, but less stable, more reactive, and it gets duplicated from RNA. And I think that the important thing about it, one of the important things about it is that it actually gets repeatedly copied, right? So that the level of the amount of expression of that um, the genes in RNA impact the behavior of the cell. So that could be because it affects the behavior or the expression of other genes. It could be because it signals to other places in the cell or outside the cell. Um, and it also, the RNA is sort of the mechanism by which proteins are created in the cell. And I think that when I previously understood until like the sort of modern era, when I like, when you learn this way back when I was learning it in college or whatever, I learned, I just had this concept of like these abstract things and they're proteins and, they're just like these strands and they kind of fold. And like, I didn't really get what they like, how that like sort of fits into like creates all these like structures, right? Like these enormously complex cells. So I think I want to just click on this this video really quick. So this is this is what's called an enzyme. An enzyme, excuse me, is a is a molecular machine that's created by from many proteins, right? And this particular one is called ATP synthase. It's what turns it like sort of refreshes, I think it's ATD, I think, and it turns it into ATP by adding, by like sort of um, by mechanically putting two pieces of protein together, and that putting the proteins together sort of like causes, it gives it energy that can be released later, like a spring or something, and so that like so this video, sorry. So can you guys hear that or no?
If I can hear.
Nope, we
You
can't
can?
hear it. Cannot.
You can. Oh, yeah, that's okay. So like the important thing here is that like, so these are ATPs that are going into here, right? And then um, so like this is all just a bunch of proteins together, right? And you can see there's this there's this sort of thing that's turning in here, and there's actually literally an electric motor. So you can see it down at the bottom here. So the there's like there's a membrane, and then there's like a bunch of proteins, or sorry, a bunch of hydrogen ions down at the bottom, or so protons or hydrogen ions, and they're those like sort of like sort of super energized things at the bottom, and that causes this to turn, and that causes it to refresh the ATP, right? So like this is like one example, one of the more impressive ones, but still like just one example of the crazy stuff that gets constructed by basically folding by like sort of putting together these proteins into these molecular machines, enzymes, plus signaling, plus other structures. So I just um, I wanted to just go over that just to be clear that there's like this this is like what's being constructed. It's not just like some soup of protein that somehow magically turns um, into like a cell and then cells into people. Okay, sorry. So that's like my okay, that's like sort of the conceptual view. And then there's so then there's like a logical view that I have that is like basically, so for DNA, um, there's basically four nucleotides. This is a kind of an approximation actually, but it's more or less there's four nucleotides. So, and they have names, but you just remember ATCG, and they kind of they fit together, like I think A and T and C and G fit together, so they're called base pairs. But they can also they can like bind to each other next to each other. So you can just have these from a conceptual standpoint, you just have a long sentence with four letters in them, right? So there's four tokens, A, T, C, and G, and then you just have a sequence of those. And that's what code, so those sort of are in your long-term storage, and then DNA gets transcribed into RNA, which is basically the same, but the T is a U in RNA, and so that has different chemical properties. It's less stable, meaning, and so that allows it to be more, and it doesn't turn into a double helix, so that it allows it to be more easily like chopped up and shuttled around, and like it's just more convenient for transcribing. So that what happens is DNA gets copied into RNA, you get these like sequences, and then, shorthanding quite a bit, the RNA turns into, there's like sequences of three RNAs, and those are called codons, and the codons attach to amino acids. And so that these codons, sort of like these patterns of codons, then turn into amino acid, like, or bind to amino acids and stick them together into these long, what's called a polymer chain. And that polymer chain is a protein. And then the proteins fold because of their chemical properties, and they turn into those amazing molecular machines. Okay, so like that's very, very like high level summary. Um, but I think the important thing for an AI engineer to know about this is that there's kind of two main things that I'm calling out here. One is that the genes are like really long, right? Like you have these long, 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 long sequences, in the one uh, 1,000 to 1 million token range, right? And then the vocab is obviously very small. So your vocabulary is four tokens, and then they actually added some other special tokens because they're just using a single byte encoding. So what do you do with the rest of the possible tokens? And so they have them like one token per species, basically. Okay, so that's my introduction to genetics. I hope that's enough for you guys to understand the rest of the discussion. Do you guys want to ask any questions?
I just want to say I love the effort put into translating things for AI engineers. I think it's very this slide quite important, and yeah, I like it.
Great, excellent, thank you. Okay, all right. So let's now. I'm gonna here's like a a gear shift with no clutch here. So I'm gonna switch to the sort of this is the main sort of mechanism by which this model is able to have long, like efficient inference on very long context. So like with that, so I'll build up to why that this mechanism works. But basically, starting in this has been research that has been developed at Stanford over the past several years. So this part of it is a little old, but um, and this refers to a previous paper that isn't in the list that we read today. So basically, they developed this what they call a hyena operator. I don't know the origin of the name, but basically as a surrogate, originally as a surrogate for attention, right? So how can we, in the sort of research question, I think was basically, how can we replace attention with something that is based on convolutions, which is kind of similar, like you've probably heard of state space models. So this is an example of a type of state space model. And so because the reason why you would want to do that is because you can convolutions are more efficient to calculate than a full attention self-attention matrix, right? So so that they can be and it's not obvious why from this diagram. I'll show you why, but that's sort of the why of it is they're more efficient to calculate. And so just to be clear, what's going on here is that you have these sort of like so you have like this is a convolution, and this is called a Toeplitz matrix, which is just a sort of like a unrolling of the convolution into a matrix, and then you have weightings on those convolutions. And so you have these pairs of like a weight plus the convolution plus a weight plus a convolution and some like arbitrary number of those stuck together, and then you can get this thing that kind of sort of approximates the behavior of attention. So this HU is basically a replacement or a surrogate for attention. But and it's important to note that there's two ways to represent there's probably more, but like there's at least two ways to represent this convolution. One is as this Toeplitz matrix, but the other is this quote unquote, they call it implicit parameterization, which means is think IIR filter, if you're familiar with those, or recurrent model, if you're familiar with those, where you're taking the previous state and you're just calculating an update rather than having all of the updates sort of written out for you. Good so far? Okay, so, and then here's the why again. Convolution is faster than attention, but note, and they actually talk about this in the paper, and that's true in the limit. It's not true at short sequence lengths, right? So if you look at hyena here, it's actually slower than flash attention up until you get to this like 10 to the 3.8, which I don't know what that's probably what like about maybe 5,000 or something context length. And also notice the performance, although they claim is sort of like a surrogate for attention, it doesn't actually do as well as GTP Neo, which is a regular attention-based model, or RWKV. Hopefully Eugene's on the call. So Eugene, you're winning too.
I'm
Okay,
willing to help
good.
answer questions for this part if needed.
Yeah, please. I don't have any questions yet, but if other people do, and especially like sort of details on state space models. I know you know a lot about them in comparison to RWKV. Okay, so then this is a slide on striped hyena. So and also striped hyena and striped hyena 2, which is what they use for Evo 2. So they actually, so here in this Toeplitz matrix here, you see that like the question is sort of like, oh, well, I have this full attention block and then I have like my fully realized Toeplitz matrix. So like, how am I actually saving in memory or whatever, right? Or computation. And so like part of the answer is, well, you can perform this convolution in the frequency domain and it's faster. So that is a good answer, but also part of it is, well, actually, it's both like more efficient and more both more efficient and better results if you actually use different convolutional or these hyena operators. So this like sort of pair of like this guy plus this guy, and then you have like a chain of those. So you can do that in different ways. And so they have 3 different ways that they propose to do this. One is this short explicit, and you can, disappointingly, they didn't have a good Toeplitz matrix diagram of this, but you can see like this is basically a line here. So it's like sort of you can think of it as like a like being somewhat related to local attention, right? Where you have just a few tokens of context close by that you're looking at, and everything else is 0. And then you have this medium regularized, which is like more context in the range of a couple hundred tokens of context, and everything else is 0. And then you have this long implicit, which means I'm going to use this implicit representation. So maybe I'll have to do recurrence in this block, or maybe I will like sort of hybridize that with sort of some implicit and some explicit representation. But in any event, you have this. It has a more efficient way of calculating using FFTs. So this, even if it's like sort of fully realized, it still is more efficient than attention to calculate. So, and then so the idea is that you have this stack. So instead of having attention block, attention block, attention block, you have this SE, MR, LI, and then an attention block, and then SE, RR, MR, LI, and then like, so you kind of stack these up so that you end up with sort of your different layers calculating sort of their attention circuits in different ways. And it turns out, well, first of all, just to be a little more clear, this is like a FIR filter, 4 to 7 tokens in depth. This is like a FIR filter, and they quote unquote regularize this. So Eugene, this is actually one place where I had a question. They say that this alpha is swept across channels. I don't actually quite understand what that means. I don't know if you read the paper or not, but like, did you understand what this is?
So I think the way to view these segments, right? And this makes sense once you understand how like future state space models work, right? Is a way to visualize it as you process the tokens. So let's just say you can view this as a genetic sequence, left to right. I hope people can see my hands right now. Left to right, the whole sequence. Same thing with text as well. The way these tokens are being merged is at the start, like these tokens gets processed and gets merged together. So as we state space model built the state by merging everything together over the layers. Does that make sense? Over time and over the layers. So so what this means here, right? In this case, right, they had these first few layers that process the tokens that come in. So we're talking about 4 to 7 tokens, and then it gets regularized and then merged into other chunks of tokens. There's, let's say, 4 to 7. I can't remember what's the exact size for future state space models and the models at that time. And then it just keeps merging cascadingly. And therefore, the layers that deal with the longer implicit attention, they view the summarized information from the previous layers that then gets merged in. So hence why they show that separation, because at, at this point they were still testing and trying to figure out how to best manage long context length and for genetic modeling, and part of the evaluation was that if they just keep one state and then they keep merging in, it performed worse in the case of state space than to have it like pre-processed with the short explicit and then subsequently merge into medium and then merge into large.
Yeah. Yeah. So I guess yeah, exactly, like the pre-processing. But that's a really good way to think about it. Yeah. What I was actually asking about was this alpha parameter. Is it says in the paper that it's swept across the channels, and they don't really go into what that means. And I think I understand what channels are, is just basically the different dimensions in your hidden dimension. Is that right? So, but then I wouldn't understand. I don't really understand why sweeping this would be a good idea, or like what, or if I'm completely misunderstanding what they're saying.
I think it's just view it as it's applying that decay um as the information flows in to force the model to like just summarize it even further. Because even in RWKV we have that decay mechanism, where where by default every piece of information will be tried to be forgotten unless the model has decided that this is important to remember.
Okay. Yeah.
So so
Yeah.
so so that decay, right, kicks in to help force the model to forget things by default.
Right.
Unless
Yeah.
as part of the matrix multiplication calculations, it decides that, hey, I should amplify this signal instead. Um and and I think genetic modeling is a very good example of like understanding why recurrent models are highly favorable to like even this use case, or even sometimes can be used for text. Is because at the end of the day, right, like if you look at the genetic code, right, it's one of the cases where it's highly repetitive, the data,
Yeah.
but there is like slight changes that we need to capture meaning.
Yeah.
And if you view it from a compression point of view, we're talking like zip compression, things like, it's highly compressible.
Yeah.
And so what you want the model to actually learn is the pattern and the anomalies, the peaks.
Yeah.
So and to discard the repetitions. Uh,
Right.
I know this is not the accurate way to say it, but in this use case, right? So by default, if we discard everything towards decay, the peaks, like, hey, this is weird. Why is genome 364, for example, is A, not as
Yeah.
what the repetition should be?
Yeah.
Then the matrix multiplication will pick that value, and then it will try to like conserve it into memory.
Right.
So that's why it may sound vague, but it's more of like the decay is there by default, and how it works from there.
Okay. Yeah. Okay. Thank you. Um, okay. So let's see. So, so basically, and this is just like sort of, um, this is kind of an ablation. They they studied, okay, the multi-head attention with their, sort of like, if you just make these all multi-head attention, you train for 400 billion tokens, and then you get a perplexity of 3.09. Right. And then they looked at this LI, which is this one. This was the original design of hyena, striped hyena, and you get this point 2.87, so a bit better. And then interestingly, if you just replace these two first LIs with these SEs, you get the same basic perplexity. So these LI, so it doesn't hurt to have this more efficient, very short convolution instead. And then they found that like you do even slightly better if you stick an MR in there. So so and you know, like for what it's worth, at this, you know, in this model, with this data, like at this point in the training process, beating multi-head attention. um, and then I actually, here's another place I don't understand what this ABF positional embedding is. I stuck it in here more as a hope that someone else would explain this slide. If not, I don't think they didn't seem to think it was super important, but that like basically this it's a more scalable, you know, this ABF is more scalable and they get up to 1 million tokens. It seems one thing I was a little unclear about is why is the perplexity going down as the context length goes up? You guys, is it just data distribution difference, or I'm not sure. Did anyone pick up on that?
For genetic sequencing, that is should be expected. Mostly because, like I say, majority of the data is going to be repetitive.
Okay,
So
yeah.
once you've seen the pattern, you should be able to at least answer the pattern.
Yeah, okay, I see. Yeah, that makes sense. Okay. All right. So training uh so training here is you know, sort of like on both 7B and 40B. Notice this is log scale significantly faster than dense transformer. You know, like you can see it here, but I think maybe even easier to understand is that, you know, like one would be the same amount of time as striped hyena 2. So they beat, you know, striped hyena 1, plus they beat, they beat the dense transformers by a factor of 3 or more here, and a little more than 3 and a little less than 3 here at the long context, right? So you can, this is what we expect, but it's sort of validation of this was one of the primary design points, and they're saying we kind of met the design point. Okay, and then they get into a lot of detail. I'm not going to go into a ton of detail here, but that there's this sort of efficient two-stage block uh, um, convolution algorithm that works well for these explicit parameterizations. So like, you know, from the upper left, here's like just a toplets matrix, and you can envision this as just blocks of matrices, right? So, you know, so this is just the full toplets, you know, lower triangular matrix. I'm just making it into smaller, or I'm just chunking it into these different capital H's. And then, so what they're saying is that if you, so you have this like a convolution, could, like if it's not a full, if it's, you know, like a short FIR filter that's only L sub H long, then I have all these zeros here, right? And so that I can actually, if this is kind of short, then I can actually represent this with just two, like the diagonal H0 and then H1 on the right next to the diagonal. Right? And so this might not be clear from the diagram. I'm going to go into that, but just notice there's zeros here. So you don't need you don't need any of the H's in this lower left-hand corner. And that's sort of, so, you know, this is a pretty good example. So if you just, you know, let's say your sequence length is 6 and your filter length is 4, then you have this, you know, sort of like on the diagonal. So you have this, you can look at like this here is this, right? And then it's repeated here, right? And then you have this off, right, adjacent to the diagonal, you have this guy, and that's right there. And so that can be decomposed into this plus this, right? And so that what this allows you to do is much more efficient multiplication. So, and they, you know, study this, and it indeed significantly faster in terms of the throughput, especially in terms of teraflops per second. You know, it's basically double or more, 2.5 times, I guess, at scale. So that's a good thing. And so this can be used for definitely for the SE, the small one, and then the small explicit, and then the medium regularized as well, depending on the block size. And so like, and then this is a comparison of those blocks to other types of blocks. So this is flash attention. This SPDA is like non-flash attention, I guess, and then it compares to Mamba and then XLSTM and DeltaNet, which I'm not familiar with these two, but presumably they're state space models.
They're linear models, basically. Yeah.
Okay. Okay, so this is another point. I did not understand this diagram, so I stuck it here in case someone wants to explain it to me. Like, I get the gist of it. So this context parallelism, so basically taking your sequence of in this case genes of tokens and sort of inferring it in parallel and then sharing the sort of doing a sort of like scatter gather approach or whatever. I think this is scatter gather for the all to all to sort of like communicate the pieces that are missing. But this is at a bulk level. I have trouble understanding this diagram. This one is more, sort of obvious to me, and like fortunately we just went over the ultra scaling handbook, and this is covered um, like sort of sequence or context parallelism is covered in the ultra scaling handbook that we just went over, but like it's a little bit, I think it's a little bit simpler for convolution than it is for attention. But with that said, this seems pretty, this is basically ring attention. And you just like this overlap has to be forwarded to the next guy before they can compute. So I don't, this one I don't get. Does anyone understand this diagram?
I think to simplify it is think of it as tensor parallel. So
Okay,
basically,
yeah.
the bulk of the calculations are done in parallel on multiple GPUs, and then once again, like state space models are at the end of the day are just like compression of the states as they merge together. So done in
Yeah,
parallel, merge, done in parallel, and
I see.
that's part of the so the first part is like the first chunk in parallel and like by context length, and then after that the information is synced across, and then they get merged, and then you get your outputs.
Okay, yeah. So that I think I see now. Okay, yeah.
One
Okay,
thing that I actually do admire the state space team, their formulation on is, well, the high level parts, right? The math can be rather complicated, right? They actually design it in a way where you can reduce the operations down to a very few uh, brilliant operations that
yeah,
is extremely counterintuitive, but it's mathematically right. So they can do
yeah.
prove that this is cheap and faster way to like do those steps, even though it may seems weird.
Yeah, no, so I spent actually quite a bit of time going through the sort of the background papers here to understand how the like especially that the mapping from their surrogate attention to like these hyena operators, and like actually the math that I didn't put it in the talk. It's actually pretty understandable, and there's no like you don't need differential equations, which is like you need, if you wanted to do the sort of the continuous version, you need to use differential equations. But for the discrete version, like it's all just basic algebra to understand. I recommend if anyone really wants to understand what's going on under the hood here, you can go and check out the hyena paper. I can give a link. It's actually linked up above in the talk. Okay, so that's sort of like the machinery. Does anyone want to ask or comment on the machinery part of this? Okay, good. So they did a whole bunch of evals. I think part of that is because unlike the sort of like actual, natural language version, there are not so many benchmarks. There are some benchmarks, but so they have to. So a lot of what they do is benchmark against real world, sort of like real world data sets that 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 are like somewhat commonly used for evals or in some cases they just came up with data sets to test against. So they basically developed their own benchmarks for this study in a lot of cases. So it's a little bit hard for me to understand how well they're actually doing. This is an exception, I guess. Needle in a haystack we're all familiar with, and it looks like they do pretty well. Um, there's not any red except for this guy. There's a 7B model seems to have trouble at 1 million context length with depth at 100%. So for I don't really understand why. I didn't look into it. So, but in any event, like they seem to do pretty well. And the way that they do this is they just stick a generated 100 base pair sequence somewhere in the context, at 10% and 20% and whatever, and then they perturb it and see if that how much that impacts the sort of the likelihood of the same sequence at the end of the context. So I think this closely matches what is typically done for a needle in the haystack test, except one thing that I was questionable to me was that they used a generated 100 base pair sequence instead of using some actual out of distribution, sort of like held out sequence from a genome that is not part of the training set. Because, and I find this questionable because since it's in distribution, well, of course you're going to naturally be perturbed if if you're naturally going to have lower or lower likelihood if you perturb the needle, right? Does that make sense?
No, I think that makes sense.
So I feel like this is unless I'm misunderstanding, I don't feel like this is the right test. Like they should have used something out of distribution. Like they actually in other places they were really careful to hold out, like they held out certain viruses because I think for other reasons, but that they test whether or not like their performance on these out of distribution viruses, and it's really poor. And that's what they had actually intended that to be the design, because I think they didn't want to bring in the genetics from those viruses into the study or into the model for some reason. Or it might have been just for monitoring, but in any event, they should have used something that was held out from the distribution or from the training set, sorry. So, okay, anyway, so then here, like they have these, so one thing I love about these bioinformatics papers is their diagrams are often really, really good and really interesting. I don't want to, like, I can talk about a few of these. I think the important thing is the title here is that they're able to predict the mutational effect on like proteins and as measured in various ways on RNA, on organismal fitness, and across different domains of life, right? Like not only in humans or something. So if anyone's interested, I can go into I think I understand most of these diagrams, but I think one interesting thing to note is that all these zero shot so it's zero shot is really clear what it means in language modeling. In this case, I was a little confused about what they meant by a zero shot prediction here. And so what they're typically doing with all these studies is they're looking at what is the change, like in some cases it's the change in likelihood or the likelihood of sort of a sequence, and using that as a threshold on that likelihood or other more complicated things to make a prediction. So like in this case they're using the change in likelihood when they mutate a sequence, and they use that as an indicator of whether it's essential or not. And I don't understand the biology of why that's a good test in this case, but that's generally, you know, you can see that here as well. Here, maybe I'm not sure can you guys read this, by the way? It might be hard to read, but like the delta likelihood, you know, like sort of is used to predict whether or not it's an essential gene. Um, right. So is that kind of clear at least from a high standpoint? So they did like you can see, each one of these is a section in the paper, and they really, I think, did a very complete job of looking at, okay, we're trying to build a foundation model. So does it generalize to a lot of different use cases without being explicitly trained on those use cases? And I think they did a good job of making that claim. Okay, and then this is, you know, more of the same kind of, but enables, you know, sort of like human clinical variant effect prediction. So, and I thought one of the, yeah, so basically similar stuff, but with human clinical effects, right? So, and this is probably one of the most important use cases for this model is being able to like I was saying in the beginning, being able to predict what will this, given the specific genetics of this patient, what will their response to this medication be, or something like that, or like what will their progress, how will their specific set of mutations impact the way a disease progresses, right? Those are like super useful you know, things you can imagine. Everyone gets, you know, you get a blood prick when you get on medication. They sequence your genome if it's not already on file, and then you can, you know, pick which medication to take based on your specific genome, right? So that's like, and this is kind of enabling that. I don't know how they claim they're doing a good job. I think it'll, you know, I think it'll, whether that bears out from a clinical standpoint, I don't know, but it, like, you know, looks very promising. Okay, so a little, sorry, anyone want to ask questions about either of these two slides? Okay, we got 10 more minutes. Okay, so let me move Eugene. uh, so this is like information on how they train the model. So interestingly, you know, the way that they thought about this was how to, like, we're going to do we're going to train, you know, a base, like as is common with long context models, they do like a base training, and then they extend the context in like sort of what they're calling mid training. That they did that. The way they think about this is like, okay, what are the sort of genetic units that we can fit in, you know, a certain, like an 8K context length. And so that, you know, you have like these, I think tRNA is transcript RNA, you have certain bacterial genes, non-coding RNA sequences, which help with the mechanism of transcription and other things like that. But like things you don't have are like these sort of like larger eukaryote genes, phages, you know, sort of like these transcript domains for humans or yeast cells. So like you need the longer context. So it's interesting because I think unlike language modeling, like the things that are here and the things that are here are more qualitatively different, I think. So it's interesting. I found this to be like an interesting difference in language modeling. And this sort of shows you like the token counts for the two models. I won't go into the rest of the slide. Okay, so then there was two really clever things that they did, or like that clearly they thought carefully about the evals here. So they and this is part of a larger diagram that I can show, but that is like super hard to understand, or it's like really chaotic and would take like 10 minutes to talk through. So, but the gist of it is all captured here, where they, they train a sparse autoencoder, they extract features, and then they identify biological features that they correspond to, right? And so and they were using this partly as a way to verify that their model is actually learning biologically relevant things, right? So there's like sort of the operational standpoint that we already saw where like, yeah, we're able to predict things that are important, but this is more of a, look, sort of a conceptual standpoint. Can we learn features that correspond to, sort of like boundaries of genes and, different types of genes and other things like that. Does that make sense? So I thought this was interesting to include in an eval. I think it's something that maybe we could do in language modeling as well, now that sparse autoencoders are becoming easier to work with. And the other thing that I thought was pretty clever was that so this chromatin accessibility is like basically an important, so this is epigenetics, meaning it's like heritable differences in the way that your genes are expressed, that are impacted by the environment. So basically so it's kind of, to, I'm probably gonna, it's like a geneticist will probably like, you know, throw up if I say it wrong, but it's like basically a way that your your genome can learn about its environment, right? So that like so that it impacts how much, or how the ways in which your genome is actually expressed based on your environment, and you can pass this in some cases onto your children. Right? So it's also heritable, but it's a different mechanism. Chromatin is one of the main ones. There are others. So what's going on here is um, they uh, they basically want, in order to be able to either do, sort of like in like in computer simulation, like using their model or other things of chromatin accessibility, or actually to um, design genes that they can, use CRISPR and other additive mechanisms to stick things into the genome and be able to control this quote unquote chromatin accessibility. So what they did was they basically sampled sequences, and then they use this, like, you can just think of this as a classifier. Basically, is this region accessible or not? Um, and then they accept some and reject some, and then they just chop out the rejected part, and then like according to the criteria, and then just keep reject, and then take only the accepted part, and then generate from there on, and then just keep iterating in that. So it's sort of like a constrained generation, basically using this sort of um, this this really complex model to be the signal that decides whether or not to reject or like the constraint. So I thought this was also very clever take on constraint generation, and like sort of it got me to thinking about, okay, what besides, like, right now we have these very deterministic things that we do constraint generation with. What are the, sort of uh, more sophisticated things, and like sort of models and downstream things that you could use for constraint generation? So that's okay. So like this, again, like sort of truck driver stop, but that's the end of the slides. Does anyone want to comment, ask questions? I can review stuff. We have only 3 minutes.
There's a question regarding, do you get a chance to look into how the data collection was done? Like what was done for Evo 1, what was done for Evo 2, specifically, I guess, for the training and processes and all that.
Yeah, so they do go over that in the paper. They actually they formed a reasonably large data set Here, let me see if I can grab the paper. Let's see. So, I think there's a good table of contents. Let me look at that. But basically, the nutshell is that they had a data set that they reused from between Evo 1 and Evo 2. They curated, there's a whole data collection section. Maybe it's towards the end or in the appendix, maybe here. Inclusion, additional results. No, sorry, I can't pull it up. But yeah, so so they augmented. So the thing that stood out for me was that they added, so they used both DNA and RNA to train. So um they and they did that explicitly because they wanted to be able to model RNA sequences and protein as well, or like sort of do downstream tasks related to RNA and DNA. So, and what they did was they switched the U symbol to the T symbol, so it looks like DNA, and so that they keep the vocabulary the same. And the other thing that that was notable in my mind was that they left out certain viruses or many viruses from I think bacterial viruses out of the training set, and I actually don't recall whether that was because they didn't want to pollute the data set with genes that would be problematic, or whether it was just because it's different and we want to be able to measure the difference between in and out of distribution data. So, but yeah, they left that out. It is in the paper. It's well documented how they collected the data. I just don't remember it.
I guess, did they collect the data or did they filter existing public data sets?
Yeah.
Or
No, yeah, it's all, I mean, it would be enormously expensive to collect the data yourself. Yeah, they filtered public data sets.
okay.
Yeah.
Actually, there's one thing that I found very interesting about the. Can you scroll up to the part where they do the 8K to the larger context thing?
Yeah, that's in the beginning, I think.
The
Sorry,
you
just
had
in the
you had
other.
it in one of your slides, yeah.
Yeah,
And
yeah, I'll just go back to the next slides, I guess.
yeah.
Yeah. Yeah, where was it?
So,
Here, oh no, it's down here.
like, to be clear, I am not a genome expert. So there's some things that I found interesting in that one, which maybe someone else who's a genome expert can chime in. But what I found interesting, right, is that so in text modeling, for example, right, when we want to train on a smaller context thing, right, one of the things that we do is just chunk the data. So so like in the equivalent, right, we'll take the large Wikipedia pages, let's just say the bacteria, and we just split them into like 100 chunks, and then we just throw it into the training for 8K. But in this
Hmm.
case, from what you explained, I think you kind of said that they didn't do that. They basically just use all the all the genome sequences that were less than 8K as the training data.
That's what it
And
seemed like to me. Yeah.
that generalized. And that to me was surprising because it's like it just means that the vocabulary, like the grammar, so the equivalent would be like the English grammar here, right, is consistent enough at 8K that it even generalized to the larger context thing. And
Yeah.
the analogy in my head, right, is let's just say all these there is a basic grammatical rules of uh Anglo-Saxon languages, basically English, French, that whole category, not including Chinese and Japanese, because there's like a different category altogether. Like they share the same lineage, and so that they have some approximate equivalent. And if you just learn the early Latin languages equivalent, and then you follow down that trail, somehow it's
Yeah.
convertible. So that
Yeah.
to me was interesting because it just means that the fundamental concept is maybe at the end of the day, base pairs, there's just some meaning and rules that we haven't fully understand, and that's captured in 8K.
Yeah. Yeah. No, I thought that was really interesting too.
Yeah.
Yeah.
But once again, I'm no genome expert. I may have gotten that completely wrong.
Yeah, exactly. That's the
I'm just
same
applying
way.
language modeling thinking.
Yeah. Yeah. Awesome. Well, I think we're out of time. I don't want to hold people past the close. I'm happy to continue chatting on Discord or whatever. But thanks a lot for listening. I hope this was interesting to people. I'm super excited about genetics. I think I'm going to dig in further. I'm working with uh, somebody to actually put together a class uh, on single cell AI models and the sort of things downstream from those. So, if you're interested, let me know. I'll try to include you.
No, I actually think it's interesting because like I've seen papers on genetic modeling that is like sub 1B model size. You can
Yeah.
conduct a class to build reasonable models on a laptop.
Yeah. Well, and especially in single cell, the models are much smaller too, right? Because you're looking at gene level, not at nucleotide level. Yeah. Cool. All right, guys. Well, see you guys on Discord. Thank you very much. This was fun for me. Hope it was fun for you.
See you guys.
Yeah. Bye.